Welcome you all to the exam race. Today we are going to learn about the evolution of prokaryotes and the origin of the eukaryotic cell. So the meaning of these two terms is actually the same, the origin of prokaryotic as well as the evolution. In the similar manner, the evolution and the origin of the eukaryotic cell. We know that evolution is all about the change in the genetic material. So before going into the depth that how does the evolution of prokaryotic cell occur, let us understand what do you mean by the term prokaryotes and the eukaryotes actually. So that you can understand how does they came into the picture and their evolution and their origin we are learning right now. So pro means they are pro. They are old. Okay. And karyon means the nucleus. Okay. Means they have the old machinery, they are not as much as advanced. However, the U means new, okay, the new, and karyon means nucleus. This is something more advanced. For example, before 10 years, right, means let us say in 2014, if we talk about, we were having the phone is simply Nokia phone, Samsung C12. But as of now, the phone we are using, we have the iPhone, okay? We have the Vivo, a different form, Vivo Y56, different pattern came, the advanced machinery. So initially, same thing is there, the machinery is here, the genetic material, okay? The genetic material. So hope this is understood. So in this topic, we are going to learn the same. Let us start with the first slide. Okay. So let me, in the protocell, if we talk about the protocell, so what was the concept of protocell come and how does the concept of protocell enter into the picture? In the O'Brien experiment and as well as the Miller experiment, we were learning that, the formation of those biomolecule, okay, the formation of the biomolecule led to the formation of the macromolecule. Right? And this macromolecule led to the formation of the probionts. Right? Probionts. And those probionts was not actually the cell, these were the precursor of cell. Okay? So how does it led to the formation of, it was not very clear at that time because obviously probe is old. So 500 million years ago, and after the same, when the rain has done and water got filled, and all these biomolecules, whatever the biomolecule are there, they form from the gases, right? We were having CH4, that was a reducing environment. So H2 was there, CO2 was there, okay? So methane, H2, CO2, and one more is ammonia, okay? So these are the name of these gases, and they turn into the conversion of biomolecule into the macromolecule. Now these gases got the energy by the thunderstorm. So eventually that was not clear which cell it is because it formed from the organic molecule. So we cannot say that it is the organic or the eukaryotes. So how does it came into the picture? And how, if you can see this diagram, the protocell to prokaryote to aerobic and anaerobic. Okay, and then they are classified based upon the same. Eukaryotic is considering animals, fungi, protist, and anaerobic is plants, photosynthetic, protist and all. Obviously eukaryotic having machinery different, prokaryotic different. Now if we talk about the anaerobic, okay, here the anaerobic term. So how does it came into the picture? Aerobic came, but later I'll tell you how. Aerobic prokaryotes. So aerobic because obviously require the presence of O2, and the time we are talking, that was the reducing environment, no O2 was there. So in anaerobic condition, what happened, when this protocell, this probiont formed, this protocell was actually dipped in the water. Okay, let me change the color. So it was dipped into the water. Let us say there is one cell, it dipped into the water. Okay, there was some water. So this cell was taking their energy from the surrounding molecules, whatever the molecules was there. Okay, we have considered it. There was some molecules, they were taking the energy. So this cell was taking the energy. This is the precursor. Or coarser weights you can say. Probionts were having the coarser weights in the microsphere. So they were in the water or in the hot dilute soup, you can say, or the prebiotics too, because it was quite hot because of the, you can say, heatness was there in the earth atmosphere. So eventually they were getting their food, right? They were getting their food. Now, at the end, when they got the food, so they were dependent, right? So we can say that yes, they are anaerobic, heterotrophic, prokaryote. Anaerobic, heterotrophic. Why? There are two modes of nutrition. The first we are having, eventually let me write, the first we are having is your autotrophic. And the second we are having the heterotrophic. Okay. Now why I am saying that it is considered under the hetero? So let me rub it. Let me explain it here. It is very clear. So if I start with the autotrophic, let me tell you why I am saying that it is a heterotroph. Hetero. H-E-T-E-R-O. Hetero means more than two. So the probiome, the cell was there in the water, it was taking the nutrients, protein, glucose was there, so consuming from the same. There was no O2, reducing environment was there, so it was dependent in that case. So that's why it is a hetero. And if we talk about hetero to auto means self, autotrophic. Okay? Tropic means nutrition, auto means self. And they were of two types, chemoautotroph. Okay? And the second one is photoautotroph. So in this way, it was of two types, chemo and photo. Now chemoautotroph is a term itself indicating they consisting of the chemicals. There were different chemicals was there, right? And the organism, after taking all the organic material, they were in the need of taking the food either from the sunlight, so they cannot use directly the sunlight. So they were using the chemicals that was present at that time, a different chemical in form of, as reducing environment was there, so it is clearly understood the sulfur was there in the H2S form. Nitrifying bacteria was there. So these things were there, and in this way they were taking continuously nutrient. So the same thing is represented over here. Let me give you a big brief over the same so that this diagram can properly understood. However, the term is very clear, anaerobic, prokaryote, and the aerobic means they require O2. Why we are calling it photosynthetic and eukaryote one, we will discuss it. This term we will discuss in detail, this one, so that you can have a proper understanding. Now, let us discuss about the starting point. The random collection of organic molecule do not qualify as living material. Now here I have made the question mark. Why? Why it is not classified as a living material? Because there was no O2 environment earlier. Okay, and it was not assured that which type of cell it was. So life organized as cell require both organization and stability over time. Okay, because obviously if there is any cell, it requires some layer to form around it, that is lipid bilayer. And also based upon their mode of nutrition is also the important parameter, mode of nutrition. So these things are eventually somehow important. Protobionts, protobionts. So such as liposome form compartment, which in turn might contain molecules such as RNA capable of reaction, catalysis and replication. So there are different protobionts. So the probionts you can say, that was in the form of lipo. Lipo means forming the lipid-like layer, okay, which in turn might contain the molecule that is RNA. That is something further process you are going to learn. Selection would favor the most successful probionts based upon their replication. So same thing is there, stability, how much the stability.